warmly welcomes you to their national webinar on recent advances in chemical sciences, part 1. All the participants are requested to please mute their microphones and switch off their cameras. We will next have the inaugural address delivered by our respected principal and chief patron, Professor Shuri Sharma. May I now request Madam Principal to deliver her inaugural address, please? Madam.
Thank you, Papia.
Welcome.
Very good evening. Respected our speaker today, Professor Debasis Das, Senior Professor of Chemistry and Honorable Registrar, University of Calcutta. Respected organizers, so we are really honored that Professor Das, in spite of his very busy schedule, has agreed to deliver the address today. And respected organizers headed by Dr. Papia Ganguly, head of the postgraduate department of chemistry, Lady Brabourne College. Respected participants, our viewers today, basically teachers, students, scholars and researchers of chemistry as well as various other related disciplines, not only of Lady Brabourne College, but a number of other teaching and research institutes. A very good evening to you all and a warm welcome, no doubt about it. It's a wonderful occasion when the postgraduate department of chemistry has organized a national webinar on recent developments in chemical sciences, and the webinar is being started now. It's a pretty technical topic, no doubt about it, and you all know that science, technology, these are not at all my cup of tea. And I really will not take much time by lingering my speech in this regard. I'm not a person capable of doing that too. Actually, usually these things, these webinars, these talks, seminars, etc., these are all organized under normal circumstances in the college auditorium and during normal hours, that is from 10 to 5. But this is not a normal situation. As you all know that the COVID pandemic has, shaken the globe, the entire globe, every part of the globe, almost every part of the globe. And the human life and lifestyle, these are absolutely changed. And these have actually undergone sea changes. And under these thoroughly changed circumstances, probably webinar is the only option. I was really, while I was working in my college today, in the afternoon, I was really feeling nostalgic with my auditorium. The auditorium, my pretty long old auditorium, it has recently been renovated and reshaped. And after the renovation, probably 3, 4 or maximum 5, 6 seminars have been organized. And now it is remained silently without witnessing a number of speakers and audiences and so on. So I'm pretty certain that it's a very timely and very fruitful effort, no doubt about it. And I'm pretty certain that after the talk is over, the talk by Professor Das, lots and lots of questions will be raised by the participants today, and lots of their probable answers too are to be provided by our speaker today. And at the end of the webinar, I'm equally certain that everybody present here, everybody taking part in the webinar will be able to know a lot more new things, and a lot more new ways of explaining them, will feel more and more enriched and enlightened. I wish today's one-day-long national webinar a grand success. Thank you.
Thank you, madam, for your very encouraging words. We are in today's webinar, we are honored to have with us our distinguished speaker, Professor Debasis Dash, Registrar, University of Calcutta, and Professor in Chemistry, University of Calcutta. Professor Debasis Dash is an eminent researcher in the field of inorganic chemistry, with special emphasis on biomimetic chemistry. Am I audible to the audience? Hello? Hello? Am I audible?
Yes. You can be heard. You can be heard.
Okay. Professor Das has completed his graduation in chemistry honors from the erstwhile Presidency College, which is now Presidency University, followed by post-graduation from the University of Calcutta. He has done his PhD from the Indian Association of Cultivation of Science under the supervision of Professor Nirmalendu Roy Choudhury, continuing his postdoctoral research at National Tsinghua University, Taiwan. Professor Das's teaching career started in the year 1999. His journey from Vishwabharati University to the University of Calcutta happened in the year 2005, when he joined the university as reader in chemistry. He is currently the Registrar, University of Calcutta, as well as Professor in Chemistry, University of Calcutta. So he's managing two very important portfolios. Professor Das is an active researcher and an acclaimed teacher. His research interests include mainly modeling of active sites of metallobiocytes, bio-inspired homogeneous and heterogeneous catalysis, development of artificial nucleases and sensors, and synthesis of nanomaterials, metal organic frameworks, and coordination polymers. 18 scholars have received PhD under his supervision. He has authored more than 120 papers in prestigious journals of both national and international acclaim. He is a recipient of the prestigious CRSI Bronze Medal awarded by the Chemical Research Society of India. He was awarded in 2019. Professor Debasis Dash is a member of various international research bodies, such as the American Chemical Society. He is a fellow of the Chemical Society of India. He has delivered presentations as invited speaker in several national and international conferences, and his research papers have received more than 1990 citations with an age index of 23. May I now request Professor Debasis Dash to please deliver his presentation. Over to you, sir.
Good evening, everybody. I think I am. You can hear me? Oh, okay.
Yes.
And, first of all, I would like to thank Principal Ma'am and the organizers for giving me an opportunity on inviting in this webinar. First ever, I am getting the experience of this type of webinar, and I must wish all of you good health and safety in this pandemic situation. And today I will talk on zinc metalloenzymes, its models study and its importance. So zinc containing metalloenzymes with special emphasis on phosphatase activities and their model studies. This is the topic of my today's talk. And all of you know, in our periodic table, we have 110 or more elements, but 92 are naturally occurring. And out of these 92, 40 elements still now has observed to be involved in biological processes. And we can categorize them as bulk, trace, alter trace in this way. And as you know, the bulk essential elements are carbon, oxygen, hydrogen, nitrogen, phosphorus, and sulfur. These are all organic components. But what are the inorganic components which are present in our biological systems in large amounts, that is in bulk amounts, that is calcium, potassium, sodium, and magnesium. And trace elements we have, that is which in the range of within 10 gram, and it is more than 10 gram, that is calcium, magnesium, etc. We have 12 elements which belongs to trace elements. So iron, copper, zinc are trace metals present in our biological systems. And today's talk I will confine only on zinc. Now, what are the roles the metal ions actually take part in biological systems? Actually, metal ions takes two types of roles. One is structural and another is functional. Structural means when a metalloenzymes contains one metal, but it is not take part in the catalytic process, then it is structural. As for example, copper, zinc, superoxide dismutase. Here copper is the active site. It take part in the catalytic reaction, but zinc is not in catalytic reaction. It is necessary to stabilize the enzyme. So it is structure. And here copper is function. Another important things we have to know, what are the most important processes in biological systems. The most two fundamental processes we have, that is the production of reduced carbon compounds, that is through photosynthesis, and another is just reverse, the oxidative metabolism of reduced carbon compounds, that is respiration. And in all biological reactions which we have, there are three types of reactions are going on. One is acid-base reaction, another is oxidation reduction, and another is free radicals. So what type of reactions we used to encounter in our laboratories, all those we are getting in biological processes, nothing different. Now question is, why a particular metal ion has been chosen for a particular or by a biological process? For example, iron is used for transporting oxygen, but copper is used for another process. But how we can rationalize the selection? We can rationalize the selection by considering these 4 points, that is basic fitness, that is chemical suitability. For a biological reactions, if it is oxidation reduction type, then nature will select that one which will be redox active. But if it is hydrolytic type, then definitely nature will not select copper, iron. It will eventually select nickel, zinc, etc. And if two metals are equally basically fit for a biological reaction, then nature will select that one which is more abundant. And when two equally abundant elements, chemically equally suitable, then it will come to the point efficiency. And another important point through which nature selects a particular metal ion for a particular job, that is evolutionary adaptation. It means whenever nature select one, then with evolution, it will make it more competent. It never give up. In our human systems, we used to just give up if it is not required. But in natural system, it never happens. What is going on, that if it once take up, then it will be continued as much as possible. Now, my today's talk is confined on zinc. Then question comes, why zinc? If you look at the next slide, you will understand what the common people think about zinc. One patient has come to the doctor, and doctor is saying that our report has come. Then, and he also said that you have zinc deficiency. Then this patient is saying, very good, don't to mention gold and silver. So gold, silver deficiency they are thinking about, but they are not thinking about the deficiency of zinc, without which our very existence will be at stake. So it is the normal status of zinc to common people, but the researchers and the chemists should realize that zinc is very important and we should work on zinc. It is, all of you know, it is photosynthesis is the most fundamental reaction for which we are surviving. And we used to concentrate or emphasize on the particular metal ion which is involved in this photosynthesis, that is magnesium. And it is the protoporphyrin 11, which is the macrocyclic system involved in this photosynthesis reactions. But we used to ignore zinc. It is, you see, this is the simplified photosynthesis reactions, that is carbon dioxide reacts with water, produces the reduced glucose and oxygen. So these photosynthesis reactions involve carbon dioxide as the ingredient. And all of you know that is the magnesium is the compound which actually take out the solar energy from sunlight and give the fuel for this chemical transformation. But if we don't have particular concentration of carbon dioxide in this reaction, then the reaction will not proceed very smoothly. And this carbon dioxide water, when reacts, we are getting carbonic acid, and which undergo dissociation to get bicarbonate and H plus. But this equilibrium is maintained by a enzyme which is known as carbonic anhydrase. Without the presence of this carbonic anhydrase, this particular equilibrium will no longer be maintained, and our photosynthesis will be hampered and it may stop. So we are not thinking about the importance of zinc. We are always used to talk about the importance of magnesium. And if we look at the zinc enzymology, we will find that nature is using zinc in varieties of enzymes. Aminopeptidase, metallo-beta-lactamase, in so many and so forth. And just I am if you have a look on this particular slide, you will understand what is the function they are performing. And if you look at the last one, that is alcohol dehydrogenase. What does it mean? It means it is the enzyme which transfers one alcohol to corresponding aldehyde. So it is an oxidation reaction. So if we don't know how the reaction actually going on, what is the molecular level mechanism, we cannot understand the reaction, the presence of the particular metal ion in that chemical transformation. For this particular issue, that is the zinc alcohol dehydrogenase, it is a conversion of alcohol to aldehyde. It is an oxidation process. So automatically we can think about that this reaction should catalyze by any redox active metal ion like iron or copper. But nature selects zinc for this particular transformation. Why? Because if we look at the molecular level mechanism of this transformation, we will understand that this reaction proceeds with hydride transfer. So hydride transfers means hydride is a base. So we need an acid. It is not proceeding through oxidation reduction way. It is proceeding through acid-base reaction way. So this is the unique example that why we have to know the exact mechanism in molecular level to understand the chemistry involved in biological reactions. And now I am going to talk about phosphatase, as it is the main theme of my today's talk. All of you know phosphate is very essential, so on and so forth. And this particular phosphatase is an enzyme which catalyzes the conversion or hydrolysis of phosphoester bond, the corresponding inorganic phosphate, and the remaining compound present in the system. So this is very important, and this plays an important role in realizing and recycling the phosphate ions from various of phosphate substrates into the metabolic pool, which is a necessary step for many fundamental biological processes. So in order to understand biological processes, we bioinorganic chemists used to apply model study. We have two types of model study. One is speculative. When the active site structure is not very well known or is not very well assigned, then we think about, we make some speculation, and with the speculation we make a model, and this study is called speculative model. And when the active site structure is well known and we are making the mimic of that active sites, it is corroborative model study. And any model study needs one model substrate. for phosphatase activity study are usually used 4-nitrophenyl for disodium salt of 4-nitrophenyl phosphate. So question come, why this particular substrate we usually use as model substrate? Because this particular substrate is UV visible inactive, but when this phosphoester bond hydrolyzed to get back the 4-nitrophenolate anion, that particular anion exhibits a lambda band at near about 400 nanometer. So just by starting the UV visible spectra, we can understand that the reaction is going on or not. Now I will start with very small, with simple system, that is zinc complexes of sheaf-based ligands. But before to start, another point I like to mention, why in the hydrolytic reactions zinc 2 has been selected by nature. First of all, zinc is a very good Lewis acid. That is one point. Another point is that sometimes deficiency becomes strength. Zinc 2 is a D10 system, so it has no crystal stabilization energy. So it doesn't bother about the coordination number. We have several coordination number. It can possess 4, 5, 6 coordination number. And in catalysis, it is the most important issue that the center should have the ability to convert its coordination number. And zinc has that ability. At the same time, the ligands which are associated with zinc 2, since it is a D10 system and it is a borderline Lewis acid, ligand systems are kinetically labile. That is also an important issue. That's why for which the natural system is accepting zinc for various catalytic reactions. But for hydrolytic reactions, what is the most important issue is that when zinc complexes attached with a water molecule, then only it will act as an hydrolytic enzyme. And what is the chemistry behind it? The chemistry behind is that water is a nucleophile, but when it binds with zinc to center, its nucleophilicity lowered down. But what is going on? The zinc has the ability to change the nucleophilicity of the bind water by changing its coordination number and oxidation and positive charge around it. So if the coordination number lowers, the pKa value of the attached water will lower down. And in the same way, when there is positive charge at the periphery of zinc center, then its pKa value of the attached water will be lower down. That means it will convert it easily to OH, which is a better nucleophile. And that nucleophile generation is very essential in hydrolytic reaction. So actually, for those reasons, zinc 2 has been selected by nature in hydrolytic reactions. So just I'm going to my zinc complexes, which we used to synthesize in our laboratory. It is sheaf-based, very simple thing, that is condensation between aldehyde and amine. You see, in this case, what is going on in the first case, we are getting one sheaf-base, which we treated with zinc acetate. We are getting a complex where 3 zinc metal ion are there. But just by changing one CH2 group, that is adding another CH2 group, the similar second sheaf-based ligand which we are obtaining on treatment with zinc acetate, we are getting a mononuclear complex. Whenever you are synthesizing complex, then first task is to characterize the complex. And the most convenient characterization in inorganic chemistry research is the X-ray structural determination. So from the structural determination, we are very sure that the complexes which we have synthesized, one is trinuclear, another is mononuclear. Now these are the enzyme model of with which we will perform the phosphatase activity. Now we have complex and we have the substrate. The substrate is 4-nitrophenyl phosphate disodium salt. And when we will mix it, if our complex can clip the phosphate ester bond, then we will get the corresponding phenolate, and that phenolate will give a lambda max at 400 nanometer. just by studying the invisible spectra, we will understand our reaction is going on or not. Look at the time-dependent invisible spectral scan. In the both cases, the reaction is going on. But if you look at the right-hand side, it is by mononuclear complex, it is not so active, but the trinuclear complex is very active. And in order to understand why trinuclear complex is more active, mononuclear complex is less active, we may think about the mechanism. And in this case, we can apply the concept of thermodynamics. And from thermodynamics, we can realize, just look at the first delta E, that is delta E3, it is minus 5.6 kilocalorie per mole for trinuclear complex, but for the mononuclear complex it is positive. So all of you know, if it is positive, means it is not continuous. My intention is to show you that what is the mechanism through which the phosphester bond cleavage is going on. So if you have a good look on this particular slide, you will see that the phosphester bond cleavage takes place through the nucleophilic attack of water to the phosphorus atom of phosphate, and that's which I have already discussed, that the importance of bind water molecule in the gene system. Without that, there will be no possibility of hydrolysis. Now, what is our motto to study the phosphate ester bond hydrolysis? Only just to understand that it is hydrolyzing or not? Not at all. We are very much ambitious, and ambitious. What is the ambitious? We like to use our complex to cleave the DNA, and so that we can use it as an anti-cancerous agent. And all of you know, DNA is a double helix, and this double helix is composed by this way, that is, there is sugar moiety, there is phosphate moiety, and base pair. So the phosphoester bond, if our complex can cleave, then there is the possibility that our complex can hydrolyze the phosphoester bond in DNA also. If it is able to clip this phosphoester bond, there is every possibility to take part in DNA damage. And all of you know, cancer is the deadliest disease still now in all over the world. And if we can use very cheap zinc metal as the source of the drug, then the cost effectiveness will come down. And at the same time, platinum drug, which is usually used, there is cisplatin, it has several side effects, as platinum is not biocompatible. But zinc is very biocompatible, so we can use zinc as an alternative. So we have to think, we have to check whether our complex can cleave the DNA or not. And all of you know that DNA, if you look, it is a double helix, and generally it exists as a supercoiled plasmid DNA in three forms, that is form 1, form 2, and form 3. Generally it is when it is form 1, it means it is a supercoiled DNA. But if it undergo cleavage, then it will convert it to nicked circular, that is the form 2. And if it's totally ruptured, then it is linear, form 3. And we can understand whether our complex is acting on plasmid DNA or not by this experiment, that is a gel electrophoresis. Look at the left-hand side diagram. Without addition of complex, it is designated by 1 len 1 and 0 concentration of complex. You will see that there is only form 1, that is a supercoiled DNA. And when we are adding our complex with different doses, then gradually the form 2 generation increases. It implies that our complex is acting on DNA cleavage. So there is a possibility that our complex may act as anti-cancer drug. Now we are going to a little more complex system. That is, it is earlier one was sheet-based ligand, and it is manic-based. And it is manic-based. Our systems which we have chosen, there is two possibility. That is that just in the ortho position of OH group, if it is the manic-based formation takes place, it is the conventional manic product. But if in one-sided, then it is abnormal manic product. And so we have every possibility to get normal as well as abnormal manic-based ligands from our reaction protocols. And interestingly, when we are using our one-sided manic product as ligand, in presence of zinc salt, we are getting dinuclear complex. But when we are using our normal or manic, that is double both-sided manic ligand, on reaction with zinc salt, we are getting mononuclear complex. This is just due to the protonation of this site by amine nitrogen, which hinders the coming of another zinc in the compartment. So we are getting mononuclear complex. And similarly, here also we have to think about whether these complexes are actually formed or not by X-ray structural analysis. Just look at the hot tape views of the two complexes, which clearly shows that one is complex 1 is dinuclear complex, and complex 4 is a mononuclear complex. Now, this is the same UV visual spectral scan in a better way. That is, we are getting very good activity with this manic base ligand. The sheep base and manic base is two different kinds. One is imine one, one is amine one. By changing the amine-imine, we are changing the activity. And we can apply different roles of halides, etc. So we have studied different things, but I am not going in details of those roles and their kinetics. It is not our today's importance. And by analyzing all those data, we can make energy profile diagram, which our students are used to encounter in our physical chemistry books. So our research can give us various data, various ideas, and various things which we have to think about. Now we are going to a more complex system. We have the system, it is two formal groups are there. So we can bind in two ways. And if we bind with these three amines, we can make three macroacyclic ligand. This is acyclic. But in reality, what we are getting, which is very surprising, that we are getting macrocyclic system. It is an 18-member macrocycle with oxazolidine and oxazine cytidine. And if you look carefully, we are getting 4 chiral centers. So very interesting, but it is not important for today's talk. We are getting macrocyclic complex that I want to convey you. And from the X-ray analysis, we confirmed that macrocyclic complexes are formed. Now, just give a look on this particular slide. Already we have give some idea that our complexes can act to catalyze the phosphoester bond. We have the idea that these complexes can cleave or can damage the DNA, so that we can use it as a anti-cancerous dead drug. But before to do it, for the further next step, we have to apply our complexes in cell line. And after that, it is human trial, animal trial, human trial, etc. So we are just going forward step by step. First of all, we think that our complexes are active or not in phosphoester bond cleavage. Secondly, we are applying those complexes, whether it is damaging the DNA or not. Now we are applying it actual cell line. AGC cell line we are using. Left hand side, that is the A, shows the cancerous cell. So when we are using our complex in 125 micromolar, what is the scenario? Just look at the picture in V, that there is the stop of cell proliferation. It means our complex is acting on cell line also. So we are going further step. And to get your viability, we used to check its MPT assay. And from the MPT assay, we are observing that complex 3 particularly is very viable as for using it in the next step, that is animal trial or human trial. So in this way, actually drug development is going on. Now it is a very popular topic, that is metal organic framework or coordination polymer. I am now switch over from simple coordination compounds to metal organic framework, and to understand whether our metal organic framework can also used as anti-cancerous drug or not. To prepare metal organic framework or coordination polymer, we need a holder. Here is 5-azido terephthalic acid is a holder, and we have the pacer, that is the diamines, that is a bipedal system. So we have 5 bipedal systems with holder 5-azido terephthalic acid, and we are using zinc as metal ion, so that we are making metal zinc containing coordination polymer or metal organic framework. And on changing the structural features of the pacer, we can change the structure as well as for coordination motifs. Just from this slide, I would like to convey how the structural features are changing on changing the structure of the ligand system. Now we have coordination polymers, but these coordination polymers have some problems. They are not very soluble in common organic solvents, and if you use water, then there is every possibility to undergo decay. So first of all, we have to check whether it is water soluble or not. But at the same time, we have to know if its size is very large, then it will not penetrate the cell membrane. In order to make it viable in biological system, we need the highest size of 100 nanometer. So you have to make our synthesized coordination polymer to nanoscale coordination polymers. And we applied a very simple green technique, that is just by hand grinding of synthesized coordination polymer, we convert them in nanoscale coordination polymers. And we checked their size, and their size in most cases are lower than 100 nanometers. So they are a good component for using in biological study. And now I am just giving you an idea what we have done with this nanoscale coordination polymers. We performed the crash experiment with cell line. It is HCT 116 cell line. Just look at the upper column. It is 0 hours, it is 12 hours, and next is 14 hours. So at 24 hours. After 24 hours, what is the situation? The situation is it is a totally concentrated cell, means it is actually a cancerous cell. So cancer cells are growing with time. When we are using our complex or we are adding our complex in this particular HTC cell line, after 24 hours from this crash experiment, it is very clear that cell proliferation stops. So this nanoscale coordination polymer is also very good component in combating the DNA cleavage, cell growth as proliferation, and so it is a good element for further study. Now we have to understand how the mechanism tells us, or what is the mechanism through which these particular nanomaterials, coordination polymers, are functioning as anti-cancerous agent. We performed the immunofluorescence study. So if you just look at this particular slide, on the control experiments, you will see the BAX and BCL2, and just look at the lower column. When we are adding complex to the to this cell line system, what we are getting, the BAX and BCL2 formation are just different, and it means the BAX formation stops. It implies that this cell growth inhibition actually is mitochondrial dependent at apoptosis. So in this way, my actual motto is to tell you that zinc is very important in our study, because zinc, although it is not very it is a boring element for chemists because it is not colorful. But it is very much important for our survival. So we need to know how the zinc complexes or zinc enzymes in our biological systems is working. But since the biological systems are very complicated, we have to study it by simple systems, that is called model systems. And through the model system, we can understand how zinc is functioning. And our actual motto is to use the zinc complexes as anti-cancerous drug. That is also ambition just. And if we can apply it in right way, there is every possibility to use our complexes as potential drug in far future. So I am now at the conclusion. So before to conclude, I like to tell you that life has evolved from inorganic materials. And in that evolution, life has incorporated every facet of inorganic chemistry that was profitable to it. It is rightly stated by Sir R.J.P. Williams. Actually, why we are thinking about the study of these metal ions in biological systems?
Excuse me,
actually,
sir,
in order to realize the life process, it is absolutely necessary to know the chemistry of every element, not only the chemistry of carbon. So life is organic, but life is inorganic. So these are my students. I acknowledge them for their contribution. I can tell something before you. And my institute, obviously, financial assistance from CSI, UGC, and BSP. And thank you for your kind patience.
Thank you, sir, for your wonderful talk. I'm sure your talk has inspired all the students we have a very large number of students as participants in today's webinar. I'm sure there will be a lot of questions for you. I'm sure you will going to answer some of them, as many as possible. Can we start with the question answer session then, sir?
Yes.
Okay. So the first question is from Dr. Kartik Gupta, Vijay Narayan Mahavidyalaya, Hooghly. He has asked that how and where alkaline phosphatase is produced in the human body.
Could you please repeat?
Yes, sure. How and where alkaline phosphatase is produced in human body?
Alkaline phosphatase is producing in our human body.
Yes, where and how?
No, actually, I am not a biologist. I don't know how it is generating in our body, but I know that the alkaline phosphatase contains zinc, and why zinc is there, that I can rationalize. But how is it producing? That is a purely biological question.
Okay, sir. We have another question. I'll just go through it. Okay. Partho Mojumdar, he has asked, is there any specific role of the Schiff-base ligand towards the particular activity of the zinc complex?
Yes. Actually, Schiff-base ligand system is important, but on Schiff-base, it is not necessary that the Schiff-base is essential for doing the phosphatase activity study. But we have to make some complex where zinc will be incorporated. So for inorganic chemist, most common ligand system is Schiff-base because it can be easily prepared and it can be tuned in a different way. So how the different peripheral system can change the activity of the zinc in catalytic system, that can be assessed by studying the Schiff-base complexes. So Schiff-base complexes actually help us to study what will be the role of positive ion nearby it, what will be the role of one methyl group nearby it, what will be the role of the 5-member ring nearby it. So we can think about why nature selected this particular ligand system for this particular functioning, and just it is a corroborative model study type, and that's why we are using Schiff bases.
Okay. Sir, can we go to the next question?
Yes.
It is by Dr. Nupur Basu. She's from the Department of Chemistry, Lady Brabourne College. Her question is, what is the strategy behind selecting the ligands in the zinc complexes synthesized for targeting the phosphodiester bond cleavage in the DNA of cancer cells?
Yes. Actually, you have to make a gene complex which will be coordinately unsaturated, so that during catalytic reaction it may, and at the same time it must contain water molecule. It is very important, because zinc compound which doesn't have any water molecule, it will be inactive in DNA cleavage, phosphatase activity, etc. Because this reaction is going through nucleophilic attack of OH or water attached with the zinc system. And zinc can change its the pKa value of the coordinated water, as I said. And that is very important. So you have, if this your system contains a zinc with addition of coordinated water, that can help you to understand, or that complex can be used as catalyst in phosphoester, particularly phosphomonoester, and diester and triester bond cleavage is very difficult. And if you particularly ATP, it is very simple to hydrolyze as we usually think, but it is not. It is kinetically very stable. Without any enzyme, it cannot hydrolyze. So in order to understand the basic chemistry, we have to know that without the presence of coordinated water with zinc, that compound cannot be used as the model system in starting the DNA cleavage, phosphatase, or in cell growth inhibition.
Okay, thank you, sir. The next question is from Linia Mukhopadhyay. She's an undergraduate third-year student of Lady Brabourne College. Her question is, many studies have shown that hydrochloroquine forms a basal lever protection from the COVID-19 virus, not to mention its vast use in combating malaria. But usually it is prescribed to take zinc supplements along with this drug. Why is it so? What additional improvement to the defense mechanism does zinc provide?
Actually, although the exact mechanism is not yet known to us, that's why scientists are struggling to get the drug. When the exact mechanism will come out, how the COVID-19 functioning in our cell systems, then we will understand what is going on. But as I think, it is a metallo-beta-lactamase that is very important thing. It is containing a gene, and if it is, it is a drug-resistant system. Actually, if we apply one drug, that beta-lactamase activity may resist its activity. So zinc addition may help to suppress the beta-lactamase activity, and so that the chloroquine may target to the target cell. Maybe I am not very sure on it.
Okay, there's another question. This is from Noyan Banerjee. He's a UG semester 2 student from Indian Association for the Cultivation of Science. Okay, so he has asked, sir, you mentioned about using the synthetic complex as an anti-cancer drug, as it can cleave the phosphodiester bonds. My question is, if it can selectively target carcinogenic cell nucleic acid content, and if it can, then what is the basis of the selectivity which prevents it from degrading regular cell DNA? Like, I think selection between carcinogenic and non-carcinogenic. That's
exactly. And for this, you have to study with your complex with the normal cell. If your normal cell MTT assay and your cancerous cell MTT assay study is done, then you will realize whether it is affecting only the cancerous cell or the normal cell. And from that, you will get an idea that it is target specific or it is non-specific. So in every study, we have to perform our gene complexes not only with cancerous cell, but also the normal cell line. Okay, and in our cases also, we performed in normal cell and we didn't find any cytotoxicity. That implies our complexes are only active on cancer cell. Am I clear to you?
Okay. Next is Kartik Gupta has again asked you, what is the function of alkaline phosphatase in human body? If you could answer him. Like.
No, I at present I am not actually very clear about the alkaline phosphatase activity. I have to just go through it. I didn't go through it in this way. Sorry for that.
Okay. So there's another one. This is by Orko Prabholayak. He has asked, other than Schiff and Manik base, any other base can be used, and is it effective? He's
Yes,
from the University of Hyderabad. He's an MSc student.
Okay. Yes, you can just think about a bipedal system, ethylene diamine system like that. But problem is that if you make a complex with ethylene diamine or any bipedal, etc., then it may already it may not contain in water. But if you have ship-based type of complexes, then there is every possibility of formation of water molecules, and there's coordination, and water coordination is very important. And at the same time, ethylene diamine type zinc complexes are more stable than ship-based type complexes, or lability is not as much as in case of ship-based complexes. So water ligand lability and water presence are very important, and that's why we used to design zinc complexes with ship-based and Manik-based ligand. At the same time, this complex preparation is easier than preparation of other complexes, other ligand complexes. That's why we usually use ship-based and Manik-based ligand.
Dr. Aparna Sen, head of the Department of Microbiology, has written, thank you for an enlightening lecture. Her question is, while testing the complex in cancer cell lines, have you tested the cytotoxicity of the complex in normal cells? If so,
Yes.
what percentage of cytotoxicity have you found?
Of course, and without that, your work will not be accepted in a very good journal, because every journal's editor will ask you whether you have performed your experiment with normal cell or not, because cytotoxicity is very important issue. So normal cell line activity you have to study normal cell with normal cell line. Otherwise, your work will not be accepted.
Okay. There's another question by Shashi Kumar Palani. The question is, what is the buffer limit or pH maintained for DNA cleavage studies? Is it comparable with natural buffer systems?
Yes, actually, we didn't add any acid or base during these reactions. We just use our complex. And generally, this complex pH is more or less around 8, and so it is as good as our normal system.
Okay. There are no more questions in the chat box, but if anybody is interested in asking any more questions, please unmute yourself. You ask the question and then again mute yourself.
Hello. Hello, sir. This is Saugato Sarkar from Ramakrishna Mission Vivekananda Centenary College, Rahoda. Thank you, sir, for your nice presentation. Sir, I have one question. Hello.
Okay. Please proceed.
Okay. Sir, whether the nano coordination polymer was soluble during the testing of the biological activity?
No. It is not soluble. We just make some dispersion phase, and with that, little concentration is enough to study this type of systems. Okay, but as I said, without making it nanoscale, you cannot apply these coordination polymers in biological systems because size is larger in synthesized nanomaterials. But if we lower down the scale to nano resin, then it can penetrate the cell membrane. And for this, we have converted our synthesized nanomaterials to synthesized coordination polymer to nanomaterials.
Okay.
But the solubility question is not very much sorted out. Dispersion takes place and that will penetrate the cell.
Okay, okay. Oh, yes, sir. Yes, sir. I was also just thinking that. Thank you, sir. Thank you.
Okay, there's another question. What particular type of cancer can be treated by zinc-containing metalloenzyme? This is by Purbali Dash, undergraduate third year student from Lady Brabourne College.
Okay. Actually, gene containing metallo complexes which we are exploring as alternative of platinum complexes using all sorts of cancer cell lines. And still now it is not that much getting importance as the platinum complexes has. Still we are fighting because if we get a good competitor of platinum complexes, then our several ideas will be clear and several issues will be resolved, particularly cost effectiveness and biocompatibility. So, but for this we have to study a lot. It is just at the inception, you can say.
Okay. So, any more questions? If there are. Okay, so if we don't have any more questions, I would again like to thank our speaker for an excellent, lucid and enlightening talk. I'm sure all of us have been very much inspired by your talk, particularly the students who have been participating in the webinar. I would also like to thank our Madam Principal, Professor Shuli Sharkar, for her encouragement. And this webinar wouldn't have been a success without the enthusiasm of the participants. So thank you, participants, for making our first attempt at organizing a webinar from the Department of Chemistry, Lady Brabourne College, a big success. Thank you all. Thank you, sir. So
Be safe and namaskar.